Hello and welcome back. We're going to be talking about the autonomic nervous system, smooth muscle, and cardiac muscle. My name is Dr. Rebecca Borup. We'll begin with a brief overview of the autonomic nervous system. It is a part of the peripheral nervous system, which means it is found outside of the central nervous system, or at least mostly outside of the central nervous system. The autonomic nervous system regulates the contraction of smooth muscles, the contraction of cardiac muscles. It also regulates glands and adipose tissue. Anatomically speaking, the autonomic nervous system, each neuronal circuit in the autonomic nervous system is composed of two neurons, a preganglionic neuron and a postganglionic neuron. The preganglionic neuron, its soma or cell body, is located within the spinal cord or the brainstem, so somewhere within here. The axon leaves the brainstem or the spinal cord and travels some distance, either a long or short distance, depending on whether we're dealing with the sympathetic or the parasympathetic nervous system, where it forms a synapse with another neuron, the cell body being located in the what's called the autonomic ganglion. The axon would then exit the ganglion and synapse onto the effector organ, whatever that happens to be, whether it's smooth muscle, gland, cardiac muscle, or adipose tissue. This autonomic ganglion would be home to the cell bodies of several other neurons, all within the autonomic nervous system. And so those would go on to different effector organs. And so as ganglions are, it's a cluster of cell bodies found in the periphery. If we take a closer look at the sympathetic nervous system, there are some properties of the sympathetic nervous system that I want to point out to you. The first is that for the most part, the ganglion of the sympathetic nervous system tends to be relatively close to the spinal cord. We have what is referred to sometimes as a sympathetic ganglionic chain. Each of these swollen regions is a ganglion with multiple cell bodies located within the ganglion. The preganglionic neuron exits the spinal cord through T1, thoracic vertebrae 1, all the way down through L2. You don't need to memorize that for the exam. And normally the preganglionic neuron is quite short. Let me correct that there. And the postganglionic neuron is much longer, exiting the ganglion and synapsing in various areas of the body. For the example, for the one I just drew, it forms a synapse onto the mesenteric ganglion, which would be in the digestive system. You can see that we also have neurons from the sympathetic nervous system synapsing with various regions of the heart, the lungs, the parietal gland, that's a salivary gland in your mouth, and you'll notice the other salivary glands as well, and of course the eyes, specifically the pupils, and so forth and so on. Bladder, kidneys, stomach, spleen, liver, and so forth. There are a few ganglion that are actually not found within the ganglionic chain, and they are located closer to the actual organ, such as the celiac ganglion shown here, the supramesenteric ganglion shown here, inferior mesenteric ganglion there. But most of the ganglions are here in that sympathetic ganglionic chain. The sympathetic nervous system tends to be our fight or flight nervous system, but I want you to also recall that to understand that the sympathetic nervous system is also involved in maintaining homeostasis. Most people think of fight or flight as a response to an emergency situation, which is true, and many of those fight or flight responses include this lovely little gland here, called the adrenal gland. Now, the adrenal gland has two portions to it. If you zoom in on the adrenal gland, it sits right there on top of the kidney, and this outer portion of the adrenal gland is known as the adrenal cortex, and this is actually going to be responsible for secreting some very important endocrine hormones such as cortisol, aldosterone. Those are secreted there, but in the center here, what we actually have is modified neuronal tissue. These are neurons, or they at least are derived from neurons, I should say. You can see the red and the purple circles in here. Let me highlight those. The ellipses, so this is actually capillaries shown here, adding blood supply to that adrenal medulla. The blue structures are the veins, so we get a nice rich blood supply flowing through here, and that's really critical to the way that this adrenal medulla functions. The preganglionic neuron for the sympathetic nervous system, it's not drawn here, but I'm just going to kind of pencil it in. It would actually form a synapse onto the adrenal medulla, and the release of neurotransmitters at that adrenal medulla then allow for the adrenal medulla to release some pretty important signaling molecules like norepinephrine and epinephrine into the bloodstream, dumping them into the capillaries and venules there. Norepinephrine and epinephrine are also alternatively known as noradrenaline and adrenaline. The adrenal medulla is therefore a modified sympathetic ganglion, and so it's an exception to this rule up here where we see two neurons in a circuit. Here we're only seeing the one preganglionic neuron and the postganglionic modified tissue of the adrenal medulla. And here you can actually see that diagram where I actually don't have to pencil it in, where you can see the cell body here and the ventral region of the spinal cord. It is a motor neuron, so it's going to exit out of the ventral horn and then synapse onto the adrenal medulla here. The neurotransmitter that is released by the preganglionic neuron is acetylcholine, and the molecules released into the blood are epinephrine and norepinephrine. Epinephrine is released in greater quantities than the epinephrine. Sorry, correction. Epinephrine is released in greater quantities than norepinephrine. Now if we look at parasympathetic neural circuits, we're going to see, these ones are shown in blue on this picture here, but you're going to see some characteristic properties of the parasympathetic nervous system, including neurons, the cell bodies located being in the midbrain and the hindbrain, and then we also have cell bodies located in the sacral region of the spinal cord. These particular neurons have very long preganglionic axons, and the ganglion for these are very close to the organ that is being affected. And so the postganglionic structure found here is very short. So long preganglionic neurons, short postganglionic neurons, as opposed to the sympathetic nervous system where generally speaking we have short preganglionic neurons and long postganglionic neurons. Again, this is just anatomically. But I do want you to look at this and notice something very interesting and important, that most often you have different organs with both sympathetic and parasympathetic input. So in this case, the sympathetic is shown in red, parasympathetic in blue, and by and large, a vast majority of the structures in our system that are controlled by the sympathetic or parasympathetic nervous system are under what we call antagonistic control. And what that simply means is, just like with antagonistic muscles where you have a flexor and a extensor, in the parasympathetic and sympathetic nervous systems, generally speaking, the sympathetic and the parasympathetic nervous system have opposite effects at the same structure. A good example is the pupil. The sympathetic nervous system will cause the pupil to dilate, whereas the parasympathetic nervous system causes it to constrict. Now, this is not always true. You do have structures that have, for example, let me illustrate this one right here, the bronchioles, shown right here. They only have parasympathetic input. And so in those cases, they are not under antagonistic control. Instead, they're going to be under what is called tonic. So the bronchial diameter is controlled by the parasympathetic nervous system through tonic control, which involves the frequency of the action potentials. There are other glands that are not exactly opposite. So for example, salivary glands, they're not really antagonistic per se, they're more complementary. I ran out of room, but pretend that says complementary. And that the parasympathetic nervous system tends to cause watery secretions, and the sympathetic tends to cause mucosal, both of which are needed to lubricate food. You'll see again that we have multiple structures being affected by the nervous system, the parasympathetic nervous system, the bladder, the reproductive organs, the adrenal gland, and the kidneys, etc., etc. And so, again, we're going to see that, this nervous system, the autonomic nervous system, is often kind of passed over as being less glamorous, less interesting than the somatic nervous system, but indeed it is actually very, very important in terms of regulating homeostasis. One of the hallmarks of the sympathetic nervous system, the autonomic nervous system, both for the parasympathetic nervous system, is convergence and divergence. We did have a chance to talk about this in the sensory nervous system, so I'm not going to talk too much about it, except just to make sure you're comfortable with the definition. This is an example of divergence, where you have one neuron activating multiple other neurons. This is an example of convergence, where you have multiple neurons feeding into a secondary neuron. Which there's advantages to both of these types of processes. It allows for the sympathetic or parasympathetic nervous system to act, for example, as a single unit through mass activation, this one in particular. So if you have an input coming from the central nervous system, like so, and it synapses in the ganglion to several neurons, we can get mass activation of multiple structures responding appropriately to the situation. Another characteristic, and this is true of both parasympathetic and sympathetic neurons, is that they have specialized synapses. Now, if we zoom in on the ganglion, so let's kind of draw a rough spinal cord here, and we look our cell bodies in the ventral region of the spinal cord here, exits, and then we're looking at the ganglion, we're going to have your typical synapses there. And so we'll have our cell bodies here like so. But when you look at the region where the nervous system, the autonomic nervous system, these neurons synapse onto tissues, this area right here, where they hit their target structure, whatever that structure happens to be, we see a specialized synapse. So instead of what I've been drawing, the classical synapse that I've drawn, which looks something like this, what we see instead is what's called a varicosity. I want to draw your attention to this picture right here first before we look at the other one. And I want you to notice that what we have here is essentially an axon that weaves its way through, in this case, smooth muscle. And the axon continues. There's a swollen region, and in that swollen region that I am copying here, or drawing over here with these little circles that I'm putting in, those are synaptic vesicles. They contain neurotransmitters. And when the action potential travels down the axon, hits that varicosity, it causes the neurotransmitters to be dumped onto, in this case, the smooth muscle cell, activating this smooth muscle cell right here. The axon then continues. It's not the end of the synapse. It continues, and it weaves through more tissue and forms another varicosity right here. And you'll see that these weaving branches actually connect back to one another. So you have one neuron that branches and diverges, and you see several neurovaricosities, swollen regions, that release the neurotransmitters. Here's more neurotransmitter here. And again, when the action potential hits that segment, so as the action potential travels, let's do a different color. So as it travels from here to here, we get neurotransmitter released right there. Down to here, we get neurotransmitter released down there. Keeps traveling until it finds another varicosity. From here to here, neurotransmitter release. From here to here, neurotransmitter release. And each time we release the neurotransmitter, we activate the smooth muscle cell that is connected or synapsing with the varicosity. And so each of these smooth muscle cells would then be activated. These are found, again, for emphasis, they're found in both the sympathetic and the parasympathetic nervous system. You see these varicosities. And what they do is allow a large area of tissue with multiple cells within that tissue to be activated at the same time. Now in the varicosities, the release of neurotransmitter is very similar to the way it is released in other synapses. An action potential, I'll just draw in my little symbol right here, for my action potential travels down the axon until it reaches the varicosity. In the varicosity, we have calcium channels. I'll draw those right there. So calcium channels on each side. And as the calcium channels arise, they're going to open in response to the change in voltage, and calcium is going to enter into the cell. When calcium enters into the cell, it's going to bind to docking proteins and promote the fusion of the synaptic vesicles with the plasma membrane. The synaptic vesicles with the plasma membrane will dump the neurotransmitters, shown here, these little circles here, into the synaptic cleft. And in the synaptic cleft, the neurotransmitter will pass across the synaptic cleft, where it will interact with the neurotransmitter receptors you can see shown here. Now, based on what those neurotransmitter receptors are, they'll cause those receptors to open. If those receptors are, for example, sodium channels, sodium will enter the cell, creating a graded potential within the cell. If it's a muscle, it will induce contraction. If it's a gland, it'll induce the secretion of hormones or sweat or digestive enzymes or what have you. And if it's cardiac muscle, again, it's going to induce contraction. Often instead of sodium, we actually have not sodium but calcium. And we'll have a chance to talk about that in detail, especially in the heart. That's going to be relevant there. The neurotransmitter, we're going to review this in a moment, but let me just kind of program this or prime the pump, as you will, is often acetylcholine. And acetylcholine, then, as you know, is chewed up by the enzyme acetylcholine esterase, which breaks that acetylcholine into acetate and choline. And then that's reabsorbed by the cell to be used to combine with acetyl-CoA from the mitochondria to make more neurotransmitter. The parasympathetic nervous system releases acetylcholine. I should note that the sympathetic nervous system often releases a neurotransmitter called norepinephrine. And norepi is going to work slightly different from acetylcholine in terms of how it's removed from the synapse. For norepi, we have a norepinephrine reuptake pump that is going to grab this norepinephrine and put it back into our cell, where it is then broken down by an enzyme called monoamine oxidase into the building prox. I don't know why it's not just put back into the synaptic vesicle. That's not something I understand, but it's broken down and then norepinephrine is resynthesized for some reason. Okay. And that actually brings us to this part right here. So the neurotransmitter that I was showing you up here in this picture here is actually shown here. And if we zoom in, and it doesn't really matter whether it's a parasympathetic nervous system, ganglion or a sympathetic, when we're dealing with the ganglion where the preganglionic neuron synapses with the post, the neurotransmitter that is released is always acetylcholine. But when we look at the tissue, so if we're actually going out to the target organ, for the parasympathetic nervous system, most of the time, I can't for right now think of any exceptions, but maybe in the middle of the night I'll come up with one. But most of the time, maybe even all of the time, the parasympathetic nervous system releases acetylcholine. And so you'll want to remember that. Parasympathetic, postganglionic cell, releases the neurotransmitter acetylcholine. The sympathetic nervous system, on the other hand, again, at the ganglion, always acetylcholine. So at the ganglion where the ganglionic neuron synapses with the postganglionic neuron, it will release acetylcholine onto the postganglionic cell. But then if we look at the neurotransmitter that is being released, most of the time the neurotransmitter is norepinephrine. This is the most common. So generally the sympathetic nervous system, postganglionic cell, releases the neurotransmitter norepinephrine. And yes, I want you to remember that. Norepinephrine, it doesn't fit. Okay, norepi. Now, there are exceptions to this rule. I'll mention them briefly. I'm not super worried about you learning all of the exceptions to the rules, but for the sympathetic nervous system, one exception, for example, is the sweat glands. Those are regulated by the sympathetic nervous system, and in that particular case, our sympathetic nervous system. So we'll just kind of sketch in a preganglionic neuron, axon, and it's going to synapse with the postganglionic neuron. And the postganglionic neuron, the neurotransmitter that is released right here, is acetylcholine. So even though it's sympathetic, at the sweat glands it releases acetylcholine. But most of the time it's norepinephrine. Now, here's some fun. Norepinephrine is also known as noradrenaline, and it binds to a number of different receptors. Now, these receptors are located in different areas. Interestingly enough, norepinephrine has a close chemical cousin called epinephrine. In general, I'm talking very broad strokes here. In general, norepinephrine is a neurotransmitter, and epinephrine is a hormone. Broad strokes. When norepinephrine is released, there's always a little bit of epinephrine released with it. And when epinephrine is released from the adrenal medulla, there's always some norepi that is released. Okay, so that's why I say with broad strokes you can classify them as neurotransmitter or hormone, but there's always just a little bit of epinephrine mixed in, and that's just because they're so closely related chemically. There are there's two main classes with several subclasses of receptors that these norepinephrine and epinephrine bind to. They're all called adrenergic receptors. named that because norepinephrine is also noradrenaline, and epinephrine, of course, is adrenaline. And so take the adren and add it to ergic, and you've got the receptors that bind to adrenaline or noradrenaline. They are grouped into alpha, shown here, and beta, shown here. I do want you comfortable with these receptors because knowing these receptors and their locations is going to help you to understand the effects of the sympathetic nervous system. So alpha 1, let's look at alpha 1 first. Alpha 1 is found in vascular smooth muscles. What does that mean? Well, vascular means the cardiovascular system. It's the blood vessels. And those blood vessels, especially some of the arteries, all of the arteries, the arterioles, and the veins, they all have some smooth muscle in the wall of those vessels. And that smooth muscle can contract or relax, and it is controlled by the alpha 1 receptor, and that alpha 1 receptor is going to cause vasoconstriction. So let's look right here. We're going to just focus right here on vasoconstriction. That means the contraction of the smooth muscle cells, making the arteriole or the artery more narrow, which increases the amount of pressure that is being placed by the vessels onto the blood. It will reduce the amount of blood flow through the blood vessel. Now you also should notice that it's also on visceral smooth muscle. The visceral means, visceral simply means the organs. That's where the original word comes to. The viscera is usually the organs found in the abdominal cavity, and it includes things like the liver, which is actually where the gluconeogenesis comes into, and it includes other structures as well, and it will cause smooth muscle contraction. Notice that it is a G protein coupled, and you should recognize this. GQ, there's a reason I taught you all of these. So the GQ ultimately results in phospholipase C being activated and IP3 and diacylglycerol being produced, which ultimately leads to the release of calcium from the sarcoplasmic reticulum, and it's that calcium that causes smooth muscle contraction. Now look at this one right here. I want you to look at this box that I just drew a circle around, and you can see here that what we have here is a relationship between epinephrine and norepinephrine. Notice that the alpha receptor binds epinephrine with greater to or equal to affinity to norepinephrine. This is a relationship to binding and to potency. Okay? Potency. So epinephrine is more potent or equal potent to norepinephrine at the alpha 1 receptor. But here's something interesting. Let me flip over here to a blank page to kind of walk us through this visually and do a thought process here. Remember that generally norepinephrine is my neurotransmitter. And so typically speaking, if we think about, here's my axon, here's a varicosity, here's the axon again, so here's my varicosity axon. And then down here we're going to say this is my, let's say it's a smooth muscle cell. So this is my sarcolemma of a smooth muscle cell. And we're dealing with the sympathetic nervous system. How do I know that, by the way? Well, hopefully you remember that the sympathetic nervous system releases a neurotransmitter, norepinephrine. So when an action potential travels down the axon, opens those calcium channels, allows calcium into my cell, my norepinephrine is going to be released into my synaptic cleft, and sitting right here in my synaptic cleft. It's a G protein coupled receptor. Whoops, I crossed the membrane a few too many times. Who cares? And it's going to bind that, activate my alpha Q, which will bind my GTP. My alpha Q will activate my PLC. My PLC will produce IP3 and diacylglycerol. And that IP3 is going to bind to my sarcoplasmic reticulum, my IP3 receptor. Calcium is going to be released. Calcium is going to cause contraction. Where's my alpha receptor? Alpha 1, it's right there. Norepinephrine, little tiny bit of norepinephrine might be released too, and it's also going to bind that alpha receptor, and it has exactly the same effect. Does exactly the same thing. And if by some chance we have epinephrine floating through the bloodstream because it's been released by my adrenal gland, and it just so happens to diffuse into the synaptic cleft, not likely, but let's just pretend it's just kind of a renegade epinephrine molecule was released as a hormone, diffuses into the cleft, it's still going to cause smooth muscle contraction. So they both do it. Now let's go back to my figure here. Don't worry about this one. ISO is a agonist that is used. It's isoproterenol, that's its actual name, and it's used in studies of these receptors, and it's not something we need to worry about as a natural ligand. All right, now let's look at the alpha 2 very briefly. We'll switch to pink. Here's my alpha 2. Presynaptic terminals. That means it's actually going to modify those varicosities. Or the presynaptic terminals of other neurons. It's the GI protein, which we haven't talked about, but this is inhibitory. You can see it's agonist potency, inhibits the release of neurotransmitter. So this is a regulatory process. Also regulates the pancreas, the platelets, the ciliary epithelium, and the salivary glands. Of these two, this is the one I want you to know, not the alpha 2, but we're really going to focus heavily on alpha 1. But I do want you to at least kind of mentally know that alpha 2 exists, and it in general regulates these other structures, pancreas, platelets, etc. All right, let's look at the beta receptors. Beta receptors, focus on beta 1. Let's look at beta 1. We're going to look at this lovely little one right here and look at the location of this beta receptor. It is in the heart, kidneys, and some presynaptic terminals, but I want you to zero in on the heart. What are we doing on the heart? Well, it binds to a GS protein coupled receptor. Activates adenylate cyclase and PKA. And you remember that again because we've talked about that, right? This is the G protein that results in PKA activity and ultimately Krebs activation. And again, we're going to kind of ignore the isoproterenol, and we're going to focus in on the epinephrine and the norepinephrine. Epinephrine, again, greater than or equal to norepinephrine, and it's going to increase heart rate. Well, let's not worry about renin release. This is going to regulate blood pressure, and I'll talk about it when we talk about the kidneys, but focus on the heart rate there. And then we also have, this one's kind of cool, beta 2, and we'll look at that one briefly. It's going to be the visceral smooth muscles, but here's the cool thing. So wait a second. We got visceral smooth muscles up here, and we got visceral smooth muscles here. Well, look at this. It's a totally different pathway than we have up here. It's not a GQ, it's a GS. And so we're going to get activation of, whoa, what are these? Calcium channels. Okay, calcium channels. And here's interesting, again, look up here. We got, for the alpha, what did I have? Muscle contraction, smooth muscle contraction. For the beta 2, what's going on? Oh my goodness, vasodilation. Vasodilation and bronchodilation. Okay. You know what activates these? Albuterol. What is albuterol? It's an inhaled substance that is used to treat asthma. And it causes bronchodilation because activation of the B2 receptor, instead of causing vasoconstriction, causes vasodilation, and instead of causing bronchoconstriction, causes bronchodilation. So this different pathway allows for a completely different response to epinephrine and norepinephrine. And look here too, it's very, very strongly, or epinephrine is very potent, norepinephrine not so much. The beta 2 tends to respond to epinephrine, the hormone. A lot more than it's going to respond to norepinephrine. And so I wanted to point that out, and the reason I'm bringing all of this up and asking you to know about all these receptors is because what this gets into is something I've talked about before, but this idea of a tissue-specific response, where we have a ligand. In this case, the ligand is epinephrine, or the ligand is norepinephrine. And if epinephrine binds to the alpha-1 receptor, it's going to cause smooth muscle contraction and vasoconstriction. If epinephrine binds to the beta-1 receptor, it's going to increase heart rate. If epinephrine binds to the beta-2 receptor, it's going to cause bronchodilation and vasodilation. So exactly the opposite of what it does on the alpha receptor. So two different responses based on the receptor type, and they are opposite responses. And we're going to talk about this more when we talk about blood vessel control and controlling blood flow. But this is a pretty important idea to understand and to really realize and look at. Beta 3, not going to worry about. It controls the adipose tissue and lipolysis, so it's kind of important and cool. Lipolysis, the breakdown of fats to produce fatty acids, which the liver can then use to produce glucose. But you guys have enough on your plates. I mean, alpha 1, alpha 2, beta 1, beta 2, they're hard enough to keep all that straight, right? All right, so looking at the adrenergic receptors and talking more about what these receptors are, let's kind of take a look at these then. Here's an example of the sympathetic nervous system and how this is going to work. So my preganglionic neurons shown here. Notice that they are going to release acetylcholine at the ganglion right here. Now look at these. I need you to focus on these. What are the receptors found at the ganglion for the sympathetic nervous system? They are nicotinic acetylcholine receptors. Where have you seen those before? These are the receptors that are also found in skeletal muscle. They allow sodium. They are a sodium channel that is activated by the acetylcholine neurotransmitter. And so that nicotinic receptor binds to the acetylcholine, triggers the release of norepinephrine from the postsynaptic cell. If we're stimulating the alpha-1 receptor, we get vasoconstriction. Notice it's in the viscera and the skin. Now this is tissue specific also, but it's also an example of compartmentation. Where are those alpha-1 receptors found? They're found in the skin, the blood vessels that are in the skin, the blood vessels that are in the viscera. Now let's look at the beta-1 receptors again. Once again, we're going to have nicotinic receptors released or being activated in the ganglion, letting sodium in, creating a graded potential in this cell that's going to lead to an action potential at the axon hillock, which is going to lead to the release of neurotransmitter norepinephrine, and that neurotransmitter is going to activate the beta-1 receptor, and that's going to increase heart rate and something called contractility, which is the strength of contraction. Same deal, acetylcholine binding to nicotinic receptors, triggering the release of norepinephrine, stimulating beta-2 receptors, and that's going to cause dilation of the bronchioles and the blood vessels, and where? Lungs. And so we get these responses. And then, of course, if I have epinephrine sneaking in here, either because we've got a little tiny bit of it released here, or because it just somehow by chance comes in through the adrenal system, we're going to be activating these same receptors. And remember that epinephrine has a much higher potency at that beta-2 receptor, which is life-saving, can be life-saving. Now, cholinergic receptors. You should be aware of cholinergic receptors and cholinergic neurons. A cholinergic neuron is a neuron that releases the neurotransmitter acetylcholine. And so here, this is a diagram of the nicotinic receptor, exactly the same as it is found in the skeletal muscle. Please note that the potassium does sneak out, but if you'll recall, the sodium tends to overwhelm that potassium. So we have more sodium that leaks in, and this is again due to, in part, the equilibrium potentials for sodium and potassium and the driving force that exists for sodium versus potassium. So even though, yes, we do have some potassium exiting the cell, the inward flow of sodium is by far the most important. That gives us a depolarizing graded potential. And if that potential is large enough to hit our threshold voltage of minus 55 millivolts, then I'll put a question mark because it's if, then we get excitation, which is the action potential. Fun stuff, blocked by the drug curare. That's actually a question in your lab manual specific to the skeletal muscles. Okay, curare will cause paralysis because it blocks these nicotinic receptors. If I don't get activation of the nicotinic receptors, I don't get a graded potential, I don't get action potential in the skeletal muscle, I don't get muscle contraction. But it also works on the autonomic nervous system because it will block the activation of these nicotinic receptors in the preganglionic or in the autonomic ganglion. Okay. Now, another type of receptor that you should know that's a cholinergic receptor, meaning it binds to acetylcholine, is this receptor called the muscarinic receptor. This is a G protein coupled receptor, and it's going to, interestingly enough, lead to hyperpolarization because it activates potassium channels. That's one possibility, and so this is going to lower heart rate. This would be what we would see in the parasympathetic. And this is actually going to happen in the post-ganglionic. Sorry, I scribble. I'm not very post-ganglionic neurons. And it depends on the neurotransmitter, and it depends on the G protein. Because we can also have the opposite effect. We can have excitation where we actually inhibit this potassium, so we don't get the hyperpolarization. We instead get depolarization because we activate a sodium or potassium channel. What makes a difference? Well, are we doing alpha I? That's the inhibitory one. I didn't really teach you that pathway, but, or, whoops, that doesn't look like an alpha. Try that again. Alpha S. Okay, and so being aware that both of these things can happen is really quite important. Muscarinic receptors are blocked by the drug atropine, which you should remember from our pharmacology lab in class. Atropine is also used in the eyes to cause the pupils to dilate. Okay. So basic function of the autonomic nervous system, I've talked about this briefly already, so let's just kind of get into this idea. In general, we're dealing with a fight or flight situation, and usually they are opposing systems. Fight or flight tends to increase various types of activities, where parasympathetic nervous systems tends to decrease it. This is very, very general, very general. It's just kind of a general idea of the way things work. And so, you know, if we're going to see the sympathetic nervous system, we tend to get things that are going to be involved in increasing our neuronal activity, our skeletal muscle activity, our brain function. So we're going to see an increase in blood pressure, an increase in heart rate, an increase in respiratory rate, an increase in our ability to metabolize ATP, and so forth. But what we also see is a decrease in the digestive function. Why? You don't need to digest your food when you're running away from the grizzly bear. This is actually why often an overload of adrenaline will lead to diarrhea, frankly, or the emptying, you getting nervous and having disrupted digestive function, because because the sympathetic nervous system is kind of screwing things up there. Whereas the parasympathetic nervous system is often referred to as rest and digest, or sometimes known as feed or breed. It is involved in reproduction, the erection reflex, hence the feed or breed, not bleed. Erection reflex, also important for digestive function, so it's going to increase digestive function. And then it tends to lower blood pressure, lower heart rate, lower respiratory rate, etc. And so we tend to see these antagonistic effects as described. A great example then is the control of the heart. If we look at the heart, please pay attention to this neuron. This is our nerve. This is the vagus nerve. That should be familiar to you. This is one of those cranial nerves that you will have learned about. And it's going to exit through the medulla oblongata, or come from the medulla oblongata, and it's going to synapse onto the SA node. The SA node is the primary region of the heart. It contains what is known as a pacemaker, and it sets the rhythm of the heart. And the vagus nerve is parasympathetic, and it regulates and tells the heart rate to slow down. So activity on the parasympathetic nervous system in the form of action potentials will decrease heart rate, and that's their vagus nerve. So know that vagus nerve decreases heart rate. Activity on the vagus nerve decreases heart rate. On the other hand, if you take a look at the sympathetic nervous system, we have the cardioacceleratory center, which is just sympathetic. It's going to come on down here and feed the sympathetic cardiac nerve, which again synapses onto the AV and the SA node. It also synapses onto what is known as your Purkinje fibers, which again are going to control the rhythm of the heart, and that sympathetic nervous system increases heart rate. This is a classic example of antagonistic control. It also illustrates kind of a very interesting principle. I like to think about this as driving a car. Most tissues have a brake, and I think of the parasympathetic nervous system as my brake, and it has an accelerator or gas pedal, which I think of as my sympathetic nervous system. So if you're driving a car and you want your car to go, especially modern cars that have these computer systems, if you're even just parked, a lot of times to get forward momentum, all you have to do is take your foot off the brake. So to increase your heart rate, in this case, the first step, step 1, take foot off the brake. And then step 2, put your foot on the accelerator. And with the sympathetic nervous system and parasympathetic nervous system control of the heart, what you need to understand is that the sympathetic, or this sinoatrial node by itself, if I took, I'm going to rip these nerves off, bye-bye nerves, they're gone, and just look at heart rate with only the sinoatrial node driving the rhythm of the heart, my heart beat would be approximately 100 beats per minute. SA node only. But now let's assume we haven't brutally ripped those nerves out, and now we have activation of the parasympathetic nervous system. Well, as the action potentials from the parasympathetic nervous system arrive, it slows down that heart rate to approximately 70 beats per minute, and we just end up getting a steady rhythm of action potentials, keeping our heart rate at its resting pace. But now if we want to increase our heart rate, if we stop, we reduce the number of action potentials from the vagus nerve, our heart is automatically going to increase to 100 beats per minute just by virtue of reducing vagus, not that kind of vagus nerve activity. We're automatically going to go up to 100. But then if we want something more than 100, let's say we're working out, and maybe we're on the treadmill and we need our heartbeat to be, I don't know, 130 beats per minute. La la la. Okay, 130 beats per minute. What we do is we've already taken our foot off the brake. Now we're going to put our foot on the accelerator. We're going to increase the sympathetic cardiac nerve activity. And so we regulate heart rate in that way. And in many cases, antagonistic control works something similar. We need to take our foot off the brake first before we put our foot on the accelerator. Antagonistic effects are not the only way the sympathetic nervous system works, and I want to make sure you understand that. So many students kind of make that mistake of just assuming that's it. It's fight or flight or rest and digest, and that there's no other option. It's one of those two. But the reality is we often get complementary or cooperative effects, such as with salivary gland secretions. I mentioned this earlier, but we get watery secretions due to parasympathetic activity, mucosal secretions due to the sympathetic division, and then complementary effects is essentially, the watery saliva, the mucus, they both do the same job, which is to lubricate food, and help our digestive system swallow that food down through the esophagus without it getting stuck. Then there's the cooperative effects, similar but different. And the cooperative effects, they work together to meet one end goal, but it's not exactly the same. With complementary, the goal is to lubricate, which is the same. And the reproductive system, the erection reflex is regulated by the parasympathetic nervous system, while the ejaculation reflex is regulated by the sympathetic nervous system. Different purposes, or I should say, well, same purpose, reproduction, but different effects in that, with the parasympathetic nervous system, we're causing vasodilation and the engorgement of the penile tissue, so that we can get penetration during sexual activity, whereas sympathetic is going to cause the spasming of those smooth muscles to eject the semen. So they complement each other without being the same. And then, equally important, we have organs that are not controlled by dual innervation, in which case there is no antagonistic control. Instead, there's only one that is going to affect this. I've already mentioned this briefly, but I want to really drive this point home. One such organ is the adrenal medulla. And so if we were to look at the adrenal medulla, remember we have in my spinal cord my preganglionic neuron, and then instead of synapsing with a postganglionic neuron, it's synapsing with the adrenal medulla, specifically that inner region right here. And then that's going to cause the release of epinephrine into the bloodstream. Well, to increase or decrease the amount of epinephrine, the way we control that is the number of action potentials that are found on the preganglionic neuron. So if I were to diagram that, most of the time we're going to see like no action potentials. We're good. Well, that actually is not true, because most of us are stressed. So we're going to have at least a few action potentials releasing a low-grade level of epinephrine. And then, oh my gosh, grizzly bear is going to eat you. We're going to release a whole crap ton of epinephrine through this burst of action potentials from the preganglionic neuron at the medulla. Sweat glands in the skin, similar kind of idea. We've got our little preganglionic neuron, we got our postganglionic neuron, and we got this little gland in the skin. And in response to an action potential being generated, we squirt that out onto the surface of the skin, type of thing. And then whether we sweat a lot or we sweat a little is again going to be regulated by this, which is called our tonic control. For the parasympathetic nervous system, we see this in the bronchial of the respiratory system. So again, same idea. by changing the frequency of the action potentials on that single input of neurons, we get a different response. So here's a little bit of a better drawing of tonic control. So let's imagine, for if we will, I'm going to show you an example of the sympathetic nervous system and blood vessel diameter, but we're going to actually imagine, since I mentioned it, the bronchioles. So let's look here. So here, this would represent a single action. Let's diagram it. So we've got our spinal cord, we've got our parasympathetic nervous system, and that's usually a long one, and then we have a short neuron, and we're going to say this synapses with a bronchiole. Okay. If the neuronal activity that is found on the postganglionic neuron looks something like this, when we have a lower frequency, then what we're actually going to get at low frequency is going to be we'll even just erase this, bronchodilation. So my bronchiole look like that, nice wide open, lots of air can get through that. But then if we increase my frequency to this high frequency, what that's going to do is cause bronchoconstriction. Not that dramatically, but you know, that should give you an idea. So dilation is by low frequency. Of those bronchioles, high frequency constriction. Again, this is in the respiratory system, the bronchioles, which are innervated by the parasympathetic nervous system. So we've got tonic control of the bronchioles in the parasympathetic nervous system. We also see the same thing by the sympathetic nervous system in the blood vessels of the skin and the viscera. And actually the blood vessels that are also going through the skeletal muscles. Let's mention those too, because those are going to be important. But more skin and viscera, I want you to focus on that when it comes to blood pressure. We'll talk about that more when we actually hit the cardiovascular system, but remember this. And so again, if we look at this one has the alpha receptor. It's actually the alpha-1 receptor. And so if we get activation on the sympathetic neuron, this would be the postganglionic. But we would see a similar pattern on the preganglionic neuron. If we get electrical signals, we might have a normal level of dilation like so. And so that neurotransmitter that is being released by the sympathetic nervous system is norepinephrine, and that norepinephrine is binding to the alpha adrenergic receptor, which, by the way, what is the receptor up here again? Do you remember? Hopefully you do. The receptor that would be on these bronchioles is the beta 2 receptor, adrenergic. Right, the beta 2. That's a sad looking beta. Anyway, beta 2. All right, so the alpha 1 on the blood vessels. And in response to the sympathetic nervous system releasing norepinephrine onto the alpha 1 adrenergic receptor, what we're going to have is constriction of the smooth muscles. So if we get a medium level of release, each of those action potentials, remember, releasing a packet of neurotransmitters at the synapse. And of course, remember that the norepinephrine is constantly being recycled into that synapse. So we're building up some neurotransmitter without a lot of it staying there. And the synaptic cleft, then we get our average type of diameter. But if we increase the frequency, compare this frequency to this one. And you can even see, notice the amount of neurotransmitter shown in this picture. Lots more neurotransmitter in the synaptic cleft. And that means we're going to have even more of the alpha-1 receptors activated. And it's a numbers game, so we're going to have a bunch more of those activated, and we are going to get more vasoconstriction. Conversely, if we look at this example, so we're going to slow down we're going to see tremendous amount of dilation. And notice that we don't have any neurotransmitter in the synapse, and as soon as we release some, that norepinephrine pump is going to bring it right back into my neuron and get rid of it. And so we have a completely relaxed blood vessel. These are, again, unusual situations. We usually have something in between, but we actually use this process to regulate blood flow to the skin and the digestive system. And so what this actually does is it's actually pretty cool if we have, let's say, a fight or flight situation. By constricting, so fight or flight, we're running away from the grizzly bear, we're going to release a ton of neurotransmitters. It's going to look like this. And so, whoa, wait a second, lots of neurotransmitters being released in the skin and the viscera. What the heck is going on? And what's that going to do? It's going to cause decreased blood flow to the skin and to the viscera, meaning in this case mostly the digestive system and internal organs. Why is that important? Well, if you're running away from the grizzly bear, you want the blood flow to go to your muscles. And so this allows us to divert the blood flow to our skeletal muscles, taking it away from the skin. Okay? And so that's actually a really important and critical thing to understand. Now, epinephrine is going to cause a tissue-specific response. This would occur in the skeletal muscles. And if you refer back up to the chart, you're going to realize, remember that we talked about the difference between beta and alpha receptors? So in this case, it would be the beta 1, I think. The beta 1 receptors found in the blood vessels that are in the skeletal muscles. And so when it binds to the beta 1 receptor in the skeletal muscle, epinephrine actually causes the blood vessels to dilate. Meanwhile, epinephrine also reinforces the effect of norepinephrine on the alpha 1 receptors. So not only do we send blood, so by constricting the blood vessels in the skin and viscera, we divert the blood to the skeletal muscles, and here we open up those blood vessels to accommodate the blood that is coming its way, using epinephrine's effect on the beta 1 receptors. And so it's a pretty cool and very elegant system designed to help us maximize blood flow. Now, interestingly enough, I talk about this with a grizzly bear, but let me talk about this in another context. Instead of running away from a grizzly bear, let's say you hop on the treadmill. Well, we have other methods of controlling this, and it's important to understand that that's exactly what we do. Through sympathetic activation of the blood vessels in the skin and the viscera, we can actually, again, in a non-emergency situation, divert the blood to the muscles. So please don't think about it as only being grizzly bear. This is also when it comes to maintaining homeostasis. When those skeletal muscles need more ATP, they need more oxygen, they need more blood flow, they're going to be producing more carbon dioxide. It's important to maintain homeostasis, and so we're going to increase blood flow to those skeletal muscles simply by virtue of exercising. And that will also happen through the activation of the sympathetic nervous system. This is shown as epinephrine, but if we were to look at the nerves itself, the sympathetic nerves, what is it releasing? Well, it's bathing this with norepinephrine, which, remember, the norepinephrine can also bind to that beta 1 receptor. So don't forget that. And so, you need more blood flow. There's no grizzly bear, but we're just going to activate the neurons that go to those blood vessels, and we're going to open them up. When we activate the sympathetic nervous system, it's not an all or none system. We're going to go all the way back up here. Sorry, giving you whiplash here. Okay, going back up here to this figure. When we command the sympathetic nervous system to activate, we could theoretically activate every single one of these, and in a fight or flight situation, maybe we will. But more often than not, we're going to activate the ones that we need. So if we need more blood flow to, what are we going to need more blood flow to? Okay, I don't see my blood vessels here. So if we need more heart rate, if our heart needs to beat faster, but there's absolutely no reason to shut down my digestive system, then I'm just going to activate the one that goes to the heart. Where is that one? We're going to activate this one right here. Okay? And that one's going to control my heart rate, and I'm going to get a little bit of faster heart rate. But I'm not going to empty my bowels because I'm just needing a faster heart rate to maintain homeostasis. And so please understand that. We're not going to just massively activate every single sympathetic neuron in our peripheral nervous system. We're going to activate the ones that we need to activate in order to maintain homeostasis most of the time. And that's a crucial thing to really, really understand. You need to maintain homeostasis. We need to make sure that everything's working great. We can disrupt homeostasis for a temporary moment when we're in an emergency situation, but it's really about maintaining homeostasis. Parasympathetic and sympathetic nervous system, ultimately it always comes back to that homeostasis. Autonomic reflexes, we've already talked briefly about this in the sensory nervous system, so I'm not going to spend much time on it except to make sure you understand that even though in the sensory nervous system and the reflexes we talked about, we use the example of skeletal muscle reflexes, we get the same thing here. So if we have a reflex, the adrenal reflex is a great example. We see something scary, or we hear something scary. The afferent neuron carries that information. It actually would be to our brain rather than our spinal cord, and our brain would be like, ooh, I'm scared, and then it's going to activate the autonomic nervous system, the sympathetic nervous system, and that's going to come on down here and target the adrenal gland and cause a massive dumping of epinephrine into the bloodstream. Just as one example of a reflex. The autonomic nervous system is, in fact, regulated by the central nervous system. Well, of course it is, because the spinal cord is the central nervous system, but it's regulated by higher centers in the brain. We can have subconscious regulation, which is going to include regulation here in the midbrain and the brainstem. And then we also, if we end up reaching the cerebral cortex, then we actually can have conscious regulation. This is actually why we can have some ability to control a kind of a panic response. So you're anxious, you're upset, you're worried, and you do some deep breathing exercises. As just an example, your cerebral cortex is choosing to modify your autonomic nervous system. A lot of the processes actually occur automatically though, and not with conscious information. And so you end up getting control of, for example, regulation of body temperature, hunger, thirst, etc., the release of glands from the pituitary, or release of hormones from the pituitary gland, emotional responses, etc., and so forth through the process of these higher centers regulating the autonomic nervous system. Limbic system. This is the center in the brain, and it's actually not one center. It's a collection of neuronal circuits that regulate emotion. It includes a region of neurons found in the central region. It's actually buried deeper inside there, but it'll also include a little area of the brain. This isn't a very good anatomical structure. Anyway, I think it's this one. I think that's supposed to be the amygdala, and that's the hippocampus. The amygdala is your fear centers, and then you also send information through neurons up into the higher regions of the brain, like so. And the limbic system controls emotion. It's also the place that is hijacked by drugs of addiction, or any addictive behaviors. But I want to help you understand that the emotions can also affect the autonomic nervous system responses. We've all been in situations where we've been anxious and maybe we've felt faint, which would be a lowering of the blood pressure. We're sweating even though we're cold, which is activation of the sympathetic nervous system to cause those sweat glands to release fluids. Our heart's racing even though we don't need it to maintain homeostasis, because again our emotional state has activated the autonomic nervous system. And so, you know, we lose our appetite, etc. This is all types of responses to our emotional state, which is why so often digestive issues and so forth come up when somebody is struggling with anxiety disorders or other types of emotional ailments. And so just be aware that it's a very common thing. The cerebellum right here, motion sickness can affect our autonomic nervous system. And of course, I think I already talked about our cerebral cortex and conscious thought regulating it, so we can focus on, okay, this is no big deal, I can do this, I can, you know, positive self-talk to calm us down. But the converse is also true. I'm really mad, I'm just dwelling on this, and I'm just gonna keep dwelling on this and so forth, and that would actually cause our higher centers of the brain to increase sympathetic activity and so forth in our body. Long-term activation of the sympathetic nervous system, specifically with epinephrine, can lead to chronic health issues. And that brings us to smooth muscles. All right. So the reason I grouped the smooth muscles and the autonomic nervous system together is because the smooth muscles are activated by the autonomic nervous system. So we take a look at our smooth muscles. Let's zoom in on this picture over here. You'll recall that smooth muscles are these spindle-shaped cells. This should be part of anatomy for you. Let's see if I can kind of draw one of them. Here's one, fairly large nuclei. It's a little hard to see the margins of the cells there. These are the spindle-shaped cells. They're kind of fatter in the middle. This would be a longitudinal cross-section. If we're going to do a transverse cross-section, we actually see these down here. This is a cross-section of one of those spindle-shaped cells. Here's a cross-section where the nucleus was caught in the middle, and you can kind of see the smooth muscles. And we usually have a longitudinal and a transverse layer. Transverse layer would be down here in each layer of muscles. So if we look at the spindle-shaped cells and we zoom in to look at how the actin and myosin look in the spindle-shaped cells, we do not have sarcomers. No such thing as a sarcomer. Remember, sarcomer is a repeating pattern of actin and myosin, with actin radiating from the Z disc, the myosin radiating from that midline point, like so, and that would be your sarcomer. They do not exist in smooth muscles. They're not there. Instead, smooth muscles have a different arrangement. They have these structures in the cell called these dense bodies. And these dense bodies are located, here's one right here, here's another, here's another. These are our anchoring spots, our anchoring points. And the anchoring points sometimes they have these intermediate filaments running from dense body to dense body to hold them in position in the cell. But they also have our actin filaments that radiate outward, like so. And then our myosin filaments are shown right here. That's not a good color to color them in. We'll do purple. So we turn the green to purple right here, and you can see that they do overlap. There would be a binding point there. They do overlap, but when we are going to contract, that myosin is essentially going to pull the actin toward the midline still. But notice, let's look at these down here. Notice over here we have more actin fibers and more myosin fibers. But the angle is completely different. It's not linear at all. It's at an angle. And so when this contraction occurs, these up here are going to be pulled this direction, and this is going to be pulled this direction. And so we end up getting this scrunching up of the cell. So as it contracts, it's going to go from, here's my relaxed state, to my my contracted state, where it's kind of going to bunch up. I'm exaggerating for effect, but bunch that cell up in multiple directions, both longitudinally and transversely. It's going to kind of connect it and bunch it up. Here's a closer look at how those actin fibers and myosin heads interact with each other. And we actually see it where we've got these heads are going to move in that direction, these heads are going to move in that direction, which makes it even greater of a kind of a bunching up of these filaments. And it allows for a completely different type of contraction. Because of this contraction, we actually can get this peristaltic waves that occur, where you get these circular contractions around the tube of, say, the digestive system or the esophagus or what have you, rather than necessarily always contracted in this longitudinal method. Smooth muscles are what is known as single unit or multi-unit. These smooth muscles interact with the autonomic nervous system. A single unit smooth muscle has our autonomic nervous system. So this would be our autonomic neuron, and it's going to form my ganglion here and continue to travel. But I want you to notice that this is really only hitting the top layer of my cells. And so when the neurotransmitter is released from those autonomic neurons, we're going to get traction first of this top layer here, but then we're going to get a wave of contraction as it spreads from neuron to neuron. That's not a good color. We'll do blue. From neuron to neuron through gap junctions. And so the top layer contracts, and the next layer contracts, and the next layer contracts. And what's actually happening is that calcium is moving through these junctions down through the tissue. And so we get this wave of contraction that occurs. And we call it single unit because it's only being regulated by this unit here. Smooth muscles often, in single units, often display what is known as pacemaker activity, which is moderated by stretching. Pacemaker activity means that they actually will generate their own electrical signal. We'll get into a little bit how that happens in cardiac muscle. I don't really talk a lot about it in smooth muscle, but pacemaker activity is usually caused by leak channels that gradually depolarize the cell. So you do see pacemaker activities in the digestive system, for example, and we'll talk about that in more detail when we talk about the digestive system, but it does result in a rhythmic contraction that doesn't require constant input from the autonomic nervous system, but the autonomic nervous system does regulate it. Okay, and the important part of this is only a few cells actually receive stimulation from acetylcholine, or for the neurotransmitter. Then we have our multi-unit, which actually requires, look at our neurotransmitter, look at our neurons. Neurons weave in between each of these tissues, which means that every single cell can be exposed, or at least most cells are exposed to neurotransmitter being released onto the surface of their cells. And that is important because the action potential activates each of these cells in concert with one another. So rather than having a wave of contraction that descends deeper into the tissue, this is going to cause a rapid contraction of all of the smooth muscle cells present. We don't see pacemaker activity in the multicellular, or in the multi-unit muscles. Then excitation, contraction, coupling. This is just a reminder of some things you already know, but I want to zero in on these pathways. We've already talked about this. If we activate an alpha S pathway, we're going to get, this is alpha Q pathway. We're going to get activation of PLC, release of IP3, activation of the IP3 receptor, and the release of calcium. Now here we're going to add an extra layer of it to it. The ryanodine receptor exists in smooth muscle and cardiac muscle and skeletal muscle. In the skeletal muscle, you'll recall that the ryanodine receptor was opened by DHP. Okay, go back and look at that if you've forgotten. And the ryanodine receptor is a calcium channel, so when DHP opened the ryanodine receptor, it let calcium out. In smooth muscles, we still have a ryanodine receptor, shown right here. But instead of being activated by DHP, the ligand for the ryanodine receptor is calcium itself. So we end up with this interesting phenomenon where calcium that is released from the sarcoplasmic reticulum through those IP3 channels can in turn activate the release of even more calcium from the ryanodine receptor. Now, both sources of calcium, from the ryanodine receptor and from the IP3, will also go on to cause muscle contraction. And I'll teach you that mechanism in a minute. But this process of calcium binding the ryanodine receptor is referred to as calcium-induced calcium release. And we're going to see this in smooth muscle. We also are going to encounter it again in skeletal muscle. Now, I will mention that sometimes we can also get the activation of a calcium channel through an alternative pathway. You'll recall that one of the alpha S pathways can do this. You'll have to go back and look at the table that I showed you with the alpha 1, alpha 2, beta 1, beta 2, beta 3 to see that there, but it is listed there that sometimes these result in the activation of calcium channels. This is usually through the activation of a kinase that in turn activates the calcium channel. I have a tyrosine kinase receptor in here, but let's ignore that one. It's actually here because I copied and pasted from a different figure and I forgot to get rid of it. So, sorry. Anyway, moving on. Now here's the pathway. This is a signal transduction pathway. It's really important that you spend time understanding it. It is different from skeletal muscle. It helps to compare and contrast it. Recall in skeletal muscle that calcium induces contraction because calcium binds troponin. Troponin moves tropomyosin out of the way, and once tropomyosin is out of the way, the activated myosin head can bind to actin, and we get the cross-bridge cycle and contraction. In smooth muscle, we have a different process. It's a step-by-step process, and practicing is the only way to really learn it. But what happens is we end up getting depolarization of the smooth muscle depolarization of the neuron, which leads to either the opening of, in this figure, it says calcium channels from the plasma membrane, which again, going up here, that would essentially be these here. So this particular figure that I stole from McGraw-Hill shows you essentially the one circled there, but you would also have the same effect if you had the ryanodine receptor channels being opened and the IP3 receptors being opened. The point is that as calcium increases in the sarcoplasm, calcium is going to bind to a calcium sensing protein called calmodulin. When calmodulin and calcium are bound together, calmodulin goes on to activate a number of proteins. One of them is my favorite protein, CAM kinase 2. It actually stands for calcium calmodulin dependent kinase 2. Not that that matters in this particular situation, but I want you to understand that calmodulin is what is known as a promiscuous protein, meaning it binds to several targets. And in this case, calcium and calmodulin as a complex will also activate a very important protein called myosin light chain kinase. Myosin light chain kinase. And once myosin light chain kinase is activated, it is going to, in turn, it's a kinase. It's going to start phosphorylating its target protein, and its target protein is, the name says it all. The target protein is myosin light chain. So what is myosin light chain? Well, if we look at the myosin in smooth muscle, it's different from the myosin in skeletal muscle in some ways. It's a different, what we call an isozyme. It's still myosin, but its properties are slightly modified. Its protein structure is slightly modified. Nevertheless, myosin in smooth muscle is still going to go through a power stroke, but when it is activated, what it does is myosin light chain kinase attaches a phosphate to the light chain, which is this region right here. This is my light chain. When that phosphate is attached, that will cause the myosin to move into, actually let me draw this differently because I drew this incorrectly. So we'll draw it this way. This is my inactive myosin, and then when we attach a phosphate here, myosin will move essentially into what is equivalent to the cocked position, and will attach onto actin in this position here with my phosphate attached. Now, in order for myosin to go execute the power stroke, what we actually need is for that phosphate to be removed. And so the phosphate is removed. Let me, we'll just erase it. The phosphate is removed by a protein called myosin phosphatase. And when the phosphate is removed, it goes back into its forward position and it drags the actin with it. And then we attach another myosin to it, and we go, another phosphate to it, and we go back into the cocked position. So myosin light chain kinase is responsible for moving us into the cocked position through the process of phosphorylation, and myosin phosphatase, whoops, let me use the abbreviation, that's much better. Myosin light chain phosphatase removes the phosphate, allowing us to go through the cocked, or through the power stroke. Now, interestingly enough, our cells can inhibit myosin light chain phosphatase or myosin light chain kinase, depending on the situation, and we can actually remain in what is known as a latch state. We see this in things like sphincters, which are designed to maintain contraction for a longer period of time. So a sphincter, of course, is usually a ring-like structure with smooth muscle around like so, and it closes down through the contraction of those muscles and opens when those muscles are relaxed, and that's regulated by the nervous system. But it actually remains in this state for a long period of time, and the reason is it inhibits that myosin light chain phosphatase so that we end up in this latch state, which is this forward state bound to and so my muscle remains contracted. It's similar in concept to rigor state, but we don't call it rigor. Okay, it's not rigor, it's latch state. And so that can allow us to maintain this long-term contraction that occurs. Now, the contraction of smooth muscle can be graded because we're not dealing with action potentials here. Instead, we're dealing with the flow of calcium into the cell, either from the sarcoplasmic reticulum or from the extracellular fluid. We end up with graded contractions. If we have a lot of calcium in the cytoplasm, we get more activation of the myosin light chain kinase and stronger contractions. If we get less activation, less calcium, we have less activation and less cycling of that cross-bridge cycle. Relaxation is going to be similar in concept to the relaxation in muscle, in that we basically need to get rid of the calcium. Once calcium is gone and it's no longer there, then calmodulin is going to dissociate from myosin light chain kinase, which will then go back to its inactive state. And then at that point, myosin phosphatase can just strip all the phosphates off of myosin, and myosin goes back to its relaxed state, and the muscle goes back to its relaxed condition. Almost done. Well, almost is relative. The last thing I wanted to talk about is the cardiac muscle. And cardiac muscle, as you know, is striated, which means anytime you see striations, you can be sure that there are sarcomeres. That's what the striations actually are, is they're the light bands and the dark bands that are formed by the arrangement of actin and myosin into those patterns that we see in skeletal muscle. Cardiac muscle is not quite as big as skeletal muscle. It's thinner. The cells themselves are thinner. They're also branching, and they have these interesting structures called these intercalated discs. These intercalated discs contain gap junctions. And if you think back to what you know about cell adhesions, gap junctions are essentially ion channels that connect the cytoplasm of one cell with the cytoplasm of another cell. So this would be cell 1, this would be cell 2. And they allow for the very rapid movement of ions. In this case, the ion we're interested in is calcium, and it's going to move in between the cells from cell 1 to cell 2 very, very rapidly. That's actually how the single unit Smooth muscles actually work as well. So we're going to talk about how we regulate the contraction of these cells then. And to understand that, we're going to actually flip over to my whiteboard here. And I'm going to walk you through kind of the big picture before I go back to my slides. So first thing I need to teach you is that in the muscle tissue of a cardiac cell, there are two types of fibers, two types of cells. The first and most abundant is what I'm going to call the contractile myocardium. That's the tissue, or we could say myocardiocyte. And this cell is designed, its purpose is to generate force. That's the major type of muscle found in the heart, of myocardium found in the heart. But there is another type of tissue, less than 2% of the heart muscle contains what is known as pacemaker myocardium, or the other name for it is autorhythmic cells, or myocardium. And I'm going to teach you about action potentials here, and I'm going to teach you about the action potential of the autorhythmic cell, even though it's less than 2% of the heart tissue. It is going to be very, very important to generate contraction in the muscle. We'll actually revisit this in the cardiovascular system, but I want to teach it to you now so you can understand how we're going to control muscle contraction in the cardiac system. The pacemakers, by the way, we find this type of pacemaker myocardium in what is known as the sinoatrial node, the atrial ventricle node, and the Purkinje fibers. All of these have pacemaker abilities. A pacemaker is a cell that generates its own action potential. It doesn't need input from the nervous system at all. It can be modified by input from the nervous system, but it doesn't need it. And so we're going to look at the pacemaker in the SA node. So here's the basic strategy behind that pacemaker. And again, I'm going to revisit this in the cardiovascular system, but I want to teach it to you now. If we look at the action potential, we're going to have milliseconds on my timeline. On the bottom there, we're going to have millivolts. So membrane potential. On my y-axis, and the myocardial cells, the pacemaker cells themselves, actually have a resting membrane potential not of -70. Instead, their resting membrane, actually they don't have a resting membrane potential, but we're going to start out at -60 millivolts. Now, they don't have a resting membrane potential. What does that mean? Well, a resting membrane potential is stable. Homeostasis is being maintained, and we have stability there. In the pacemaker cells, there's no such thing as a stable membrane potential. Instead, what ends up happening is the membrane potential creeps upward, the cell depolarizes, membrane potential creeps upwards. Hey, stop. Membrane potential creeps upwards, cell depolarizes, and on we go, and those are really ugly curves, but please bear with me. On and we go for the life of the cell. Each of these are action potentials, and they're rhythmic, hence the autorhythmic cells, and they happen automatically. In the SA node, they're going to happen at a rate of 100 times per second. Now, stimulation from the parasympathetic nervous system can actually slow these down. So it'll slow the rhythm down to, say, 70, or whatever resting heart rate happens to be for you, per second. Or, so that's the parasympathetic. And the sympathetic can increase this. So it can increase this quite dramatically. So we can be just a little bit to 130 to whatever our maximum heart rate happens to be. And each of these pulses will lead to a beat of the heart. Okay, a beat of the heart. And so this is our autorhythmic cells. Now what causes this? This portion right here is caused by a leak channel. Specifically a sodium leak channel called this is my preferred name, and it is one of the names it's called by, but the I-funny channel. And I prefer it because it's easier to remember. The other one, I believe it's HNC. But see, I can't even remember it. So I go with I-funny because, you know, that's the easier one for me to remember. And what it does is it allows sodium to leak into the cell, and it leaks into the cell faster than the sodium and potassium ATPase can deal with it. So as sodium leaks into the cell will gradually depolarize, and then we have another channel, a voltage-gated calcium channel. And the threshold voltage for that one is going to be about approximately minus 40 millivolts to minus 35 millivolts, somewhere in that range. And that is then what happens here is we end up with this portion here is actually the only action potential of all the action potentials that I'm going to teach you this semester. This is the only action potential where the rising phase is due to those voltage gated calcium channels. The only one. And then this other portion here is actually due to voltage gated potassium channels. So we have our voltage gated potassium channels in here too. And like the potassium channels in the neuron, these are slow to open. They actually start opening somewhere down here, probably right there. But they open slowly, and so they don't maximally open until the peak of the action potential, and they will bring us, they're trying to reach that minus 90, but they're not going to make it. They'll get all the way down to minus 60 before those leak channels start pouring sodium back into the cell. Now the leak channels never really close, so they're going to be open in this region as well. Okay, but it's just a matter of being overpowered by potassium for the falling phase and so forth. So those leak channels are really quite important to this process, and it's the reason that this is an automatic process. And the parasympathetic nervous system, the way it works is it modifies those leak channels so that they are less leaky, making the rising making that in fact, let me go ahead and draw it over here. If I have a less leaky channel, I'm going to exaggerate, but see how the steepness of the curve, so we're not going to reach that minus 40 until much later. Where on the other hand, my sympathetic nervous system is going to make my channel much more leaky, so I'm again exaggerating the shape of the curve, but again I'm going to hit that action potential a lot sooner. And so we can regulate the rhythm of the heart in this manner. Now, in the skeletal muscle fibers, the contractile fibers, we're going to have a different action potential. Yay! And then I'm going to explain to you how this actually results in contraction. But first, let me give you some orientation. So let's go back to this drawing that I did. Here's my gap junction. Just kind of orient yourself. Okay. Like so. So we're essentially sending the signal to spread through my intercalated discs. On and on and on, my intercalated discs from cell to cell to cell to cell. Now let's designate this one as my autorhythmic cell. So in the plasma membrane here, I'm going to get repeatedly the automatic generation of my action potentials here, over and over and over again. Now remember that in the rising phase of this action potential, right here, we have calcium entering into my cell through voltage-gated calcium channels. That calcium ion is going to spread from my sarcoplasm of the one cell into the sarcoplasm of the other cell. And that's really important because it's going to be this calcium that is going to be the source of the voltage necessary to trigger an action potential in my contractile cell. Now, what is that action potential going to look like? Well, let's draw it down here. In my contractile cell, we do have a resting potential of about minus 85, if I remember correctly. And that cell is going to remain at minus 85 until calcium floats into that cell, the sarcoplasm, from the autorhythmic cell. At which point that calcium is going to cause a rapid depolarization, and it's going to trigger the opening of voltage-gated sodium channels, which is going to give me a very rapid rising phase. Now these channels inactivate just like the channels in the neurons. They're going to inactivate at about plus, actually it's a little lower. I think it's about plus 20 millivolts. And when that happens, the cell is going to begin to repolarize because we also, in fact, let me erase this because this is going to get in the way for me. The cell is going to repolarize because we also have the opening of voltage-gated potassium channels. And once again, those channels are going to open kind of down here, but remember they open very slow, so we're not going to reach the peak of voltage-gated potassium channels, or the maximum opening of the voltage-gated potassium channels until the peak. And the cell is going to start to repolarize. But in my contractile cells, if we were to look at this, let me kind of diagram, well, I'll diagram it a little differently somewhere else. Well, we'll do it here. We're going to put a T-tubule right here. Okay, so that T-tubule right here is going to contain a specialized calcium channel that is going to open up and allow more calcium into my cell. So my calcium channel found here in my T-tubule will now allow calcium. So not only do we have calcium that floated in from the autorhythmic cell, we're going to have calcium float in through these other channels here. We're going to have my voltage-gated sodium channels that are going to open up. Don't worry, I'm going to walk you through this again. So sodium's going to come into my cell through rising phase. Potassium's going to leave my cell for the falling phase. But then when I get to here, my opening of my voltage-gated calcium channel, I'm going to get this interesting thing happening. Potassium is going to continue to leave my cell, but now calcium's coming in and it's going to counteract that. If you look at it like this, if I were to draw the plasma membrane here, what I would actually see is potassium continuing to leave. Whoops. Potassium continuing to leave my cell at the same time that calcium is coming into my cell. And so we end up having an exchange. For every positive charge that comes out, we've got a positive charge coming in. So we'd essentially would have 2 potassium, by the way, for every 1 calcium, since the charges need to balance. And that will ultimately lead to this stage right here, which we call the plateau. And that plateau is due to both potassium leaving the cell and calcium coming into the cell. Now the rest of this is actually so that we go back to the falling phase when those calcium channels close. So now my calcium channels have closed where the blue is, and we're back to potassium leaving because these channels have closed. And that's essentially the action potential for the contractile myocardium. Let's walk through that a little bit again, and again we're going to draw, this time I'm going to draw bigger. So we have, this will represent my pacemaker cell, and this is going to be my gap junction. Obviously there would be more than one, but that's my gap junction for my pacemaker cell. So my pacemaker cell is going to generate my action potential like so. And my calcium is going to enter in through this graded potential, or through the gap junction, into my cell and depolarize my cell. Now let's look at my cell. We're going to have a plasma membrane, and in that plasma membrane we're going to have a number of voltage-gated sodium channels, which I'm drawing in red. So a whole bunch of those. And anytime you see voltage-gated sodium channels, you should automatically be thinking, aha, we're going to get an action potential. And we have voltage-gated potassium channels here. And again, if you see a voltage-gated potassium channel, you should see, aha, falling phase of an action potential somewhere, somehow. Okay? And so as the calcium comes in, it's going to provide the change in voltage necessary. We're going to reach that minus 55 millivolts in the cell, causing the opening of those voltage-gated calcium channels. And so if I were to diagram the action potential here, we're going to see the rising phase due to reaching threshold voltage thanks to the calcium that is coming from my autorhythmic cell. Now, the threshold voltage, we're also going to get the opening of those potassium channels, and again we're going to get the falling phase. But that voltage, the change in voltage, the action potential that's going to be generated, is going to travel down this plasma membrane or the sarcolemma, and it's going to enter into the T tubules down here. Now the T tubules are an extension of the plasma membrane, just like we discussed in the skeletal muscle. And also, like in the skeletal muscle, it's going to butt up against the sarcoplasmic reticulum. I'm actually going to change how I draw that because I need more room to draw what I need to draw, but understand they're very close together. And so my sarcoplasmic reticulum, okay, like so. And of course I have a bunch of calcium in my sarcoplasmic reticulum. As the action potential travels down the T tubule, and again we're going to have voltage-gated sodium channels, so scribble some of those in, and voltage-gated potassium channels, and so my action potential is literally going to travel along the sarcolemma and down the sarcolemma. And in skeletal muscles we would encounter a DHP receptor, but we're not in the skeletal muscle anymore, and so instead we're going to encounter a lovely protein called the L-type calcium channel. And this L-type calcium channel is voltage gated, and so it's going to be opened by the action potential, and not surprisingly it's going to allow calcium from the extracellular fluid into my sarcoplasm. Now in my sarcoplasm, again, remember this is not the DHP. It is a chemical cousin or a protein related to it, but it is not the DHP. So in the sarcoplasm, that calcium is going to float around, and some of it will cause muscle contraction. But others are going to other calcium ions are going to come over here and bind to my ryanodine receptor, causing the release of even more calcium from my ryanodine receptor. Now that calcium, or from the sarcoplasmic reticulum, now that calcium is going to go on to cause contraction. Now the action potential, I'm not going to continue drawing the channels, but the action potential will continue to travel through the sarcolemma, and importantly, over here we're going to have another cell, and we have another gap junction, and another contractile cell, like so, and the calcium that's being released from the sarcoplasmic reticulum, some of it's going to come over here and start the process in the next cell. Now, calcium causes contraction by binding to troponin, and the steps from this point on are exactly the same as they are in skeletal muscle. And then of course, to end the entire process, we need our calcium ATPase here in the Sarcoplasmic Reticulum, which is going to take that calcium and pump it back into my Sarcoplasmic Reticulum. We also have a pump, I'll draw it over here, at the plasma membrane that is going to move my calcium out of the cell. And that would then lead to calcium or to muscle relaxation. So let's flip back over here then and go ahead and take a look at my notes here to walk you through that process. So here's the pacemaker potential. Here's the pacemaker cell. And this portion right here, this leaky portion where the leak channels change the membrane potential, we call that the pacemaker potential. We can't call it resting because it's unstable. Threshold voltage is about -40 for those calcium channels. So those calcium channels are going to open up and cause the rising phase. And then my potassium channels are going to open and cause my falling phase. And then we're going to repeat this process. And then notice here, my electrical current will spread from my pacemaker cells through each of these contractile fibers, creating a wave of contraction and a generation of force. Here's my action potentials in my contractile fibers. The calcium channel that is found in my contractile fibers is shown here. This is my L-type calcium channel. It is similar in relationship to the DHP receptor, but it's not the same. If this were skeletal muscle, this would be tethered physically to my ryanodine receptor, but in cardiac muscle it is not, and in smooth muscle it is not. And so in smooth muscle and cardiac muscle both, we're going to rely on calcium-induced calcium release. Okay, so don't forget that. Calcium-induced, calcium released in both smooth and cardiac muscle. Excitation and contraction process is slower because we have to rely on diffusion of the calcium ions. When it's physically connected, it's a much faster process. And then here's my pump that's going to be my calcium ATPase to move calcium back into the sarcoplasmic reticulum and end the process. There's also a channel on the surface of the cell that's not shown here. Here we can see a small glimpse of the contractile cell. So remember, calcium is going to be diffusing into my muscle fiber from the pacemaker cell or from another contractile fiber, and it's going to activate these voltage-gated sodium channels. Not shown here are the voltage-gated potassium channels, but they're there. This picture's kind of a lazy picture. So voltage-gated potassium channels, voltage-gated calcium channels. If I sketch my action potential, that's going to cause my rising phase here. And the voltage-gated potassium channels causing my falling phase, and then that's going to lead to the activation of these voltage-gated calcium channels right here. These are my L-type calcium channels. L simply stands for long, by the way. And those L-type calcium channels are going to allow calcium in, and calcium is going to reactivate my ryanodine receptor. The calcium from the L-type calcium channels and from the ryanodine receptors give me my plateau, and then they eventually close, and I have my relaxation phase, my repolarization phase. And then, of course, my calcium ATPase. Notice right here we have my troponin, my tropomyosin. It's exactly the same cross-bridge cycle. You've studied this. You know this. ATP binds to my myosin head to activate it. It's hydrolyzed into ADP and inorganic phosphate. We move into the cocked position. When tropomyosin is in the way, we're just out of luck. But when tropomyosin is not, we can form a tight bond. So when calcium binds to that troponin, it moves tropomyosin out of the way, we form a tight bond here. And then when we go through the power stroke, we drag that actin with it, and we shorten the sarcomers. And that's it. That's a wrap. Okay. Now we went through that kind of fast. Hopefully that helps you. Spend some time studying this. I'm happy to go through it again with you in class if you need a little bit of a slower pace and a refresher course or whatever you need. Have a great day. Bye-bye.